We begin our journey into the microscopic realm, a universe teeming with activity and wonder. Imagine, for a moment, a bustling metropolis, a city alive with constant motion, construction, and communication. This is the world inside a human cell, a tiny universe containing everything needed for life. At the heart of this bustling city lies the nucleus, the cell's control center. Within its protective walls resides DNA, the molecule of life, a blueprint containing the instructions for everything a cell does. Imagine DNA as a vast library, filled with volumes of information passed down through generations. These volumes hold the secrets to our traits, our functions, and our very existence. Just as a city requires constant maintenance and growth, so too does a cell. Cells must replicate, creating identical copies of themselves to replace old or damaged cells. This process of replication, called mitosis, is an intricate dance of duplication and division, ensuring the continuity of life. Join us now as we venture deeper into this microscopic metropolis, exploring the remarkable processes that keep our cells, and us, alive and thriving. Mitosis, the process of cell division, is a tightly regulated ballet of molecular movements, ensuring that each daughter cell receives a complete set of genetic instructions. This intricate dance unfolds in a series of distinct phases, each marked by characteristic events. The curtain rises with prophase, a period of preparation and organization. The DNA, initially a tangled mass of genetic material, condenses into tightly packed structures called chromosomes. Imagine a librarian meticulously organizing scattered scrolls into neatly bound volumes. Meanwhile, the nuclear envelope, a membrane surrounding the nucleus, begins to break down, like the walls of a library dissolving to grant access to its precious books. As the dance progresses to metaphase, the chromosomes align gracefully at the cell's equator, forming a line as if awaiting inspection. Imagine a grand procession, each chromosome a participant in this cellular parade. This precise alignment is crucial, ensuring that each daughter cell receives an equal share of genetic material. The tempo quickens during anaphase, a period of separation and movement. The chromosomes, each composed of two identical copies, are pulled apart by spindle fibers, protein threads acting like microscopic puppet strings. Imagine a perfectly synchronized tug-of-war, with each chromosome evenly distributed to opposite poles of the cell. Finally, the curtain falls with telophase, a time of reconstruction and closure. The chromosomes, having reached their destinations, begin to unwind, reverting to their relaxed, thread-like form. Meanwhile, new nuclear envelopes form around the separated chromosomes, like protective walls rising around newly established libraries. The cell then divides into two daughter cells, each identical to the parent cell, marking the end of mitosis. This intricate dance of division ensures the continued growth, repair, and renewal of our bodies, a testament to the elegant choreography of life at its most fundamental level. Imagine DNA as a language, a code containing the instructions for building and maintaining a living organism. This language, written in the arrangement of chemical bases, holds the key to understanding the diversity of life on Earth. The DNA molecule resembles a twisted ladder with the rungs formed by pairs of chemical bases: adenine A, thymine T, guanine G, and cytosine C. These bases follow specific pairing rules, A always bonding with T, and G always bonding with C. This complementary base pairing is essential for DNA replication and protein synthesis. The sequence of these bases along the DNA molecule dictates the order of amino acids, the building blocks of proteins. Proteins, in turn, are the workhorses of the cell, carrying out a vast array of functions from transporting molecules to catalyzing chemical reactions. The process of converting the DNA code into proteins involves two main steps: transcription and translation. During transcription, the DNA sequence is copied into a messenger molecule called RNA, mRNA. Imagine a scribe meticulously copying an ancient text, preserving the information for future generations. This mRNA molecule then carries the genetic message from the nucleus to the ribosomes, the protein synthesis machinery of the cell. In translation, the mRNA message is decoded and used to assemble amino acids into a specific protein. Imagine a master craftsman interpreting a blueprint, carefully assembling each component according to the instructions. This process involves another type of RNA, transfer RNA, tRNA, which acts as an adapter molecule, bringing the correct amino acid to the ribosome based on the mRNA code. Section 4. The Molecular Symphony, Orchestrating Protein Synthesis. The process of protein synthesis from DNA blueprint to functional molecule is a remarkable symphony of molecular interactions, a testament to the elegance and complexity of cellular machinery. Imagine a bustling factory floor with specialized machines working in concert to produce a finished product. The mRNA molecule acts as the blueprint, carrying the genetic code from the nucleus to the ribosomes, the protein assembly lines of the cell. Ribosomes, complex molecular machines, bind to the mRNA and begin to read the code. Each three-letter sequence on the mRNA, called a codon, corresponds to a specific amino acid. Imagine a decoder ring translating the language of nucleic acids into the language of proteins. tRNA molecules, each carrying a specific amino acid, enter the ribosome. The tRNA molecule with the anticodon, a three-letter sequence complementary to the mRNA codon, binds to the ribosome. Imagine a lock and key mechanism, ensuring that the correct amino acid is added to the growing protein chain. As the ribosome moves along the mRNA molecule, codon by codon, the corresponding amino acids are linked together by peptide bonds, forming a polypeptide chain. Imagine a chain being assembled, link by link, following the precise instructions of the blueprint. Once the entire mRNA sequence has been translated, the polypeptide chain folds into a specific three-dimensional shape, determined by the sequence of amino acids. This unique shape dictates the protein's function. Imagine an origami masterpiece, its final form determined by the precise folds and creases. This intricate dance of molecules, from transcription to translation, ensures the production of the proteins essential for life, a testament to the remarkable symphony of cellular processes. Section 5. The Circle of Life. Navigating the cell cycle. The cell cycle, a tightly regulated sequence of events, governs the life of a cell, from its formation to its division into two daughter cells. This cycle ensures that cells divide only when needed, preventing uncontrolled growth and maintaining tissue integrity. The cell cycle can be divided into two main phases: interphase and the mitotic M phase. Interphase, the longest phase of the cell cycle, is a period of growth, preparation for division, and normal cellular function. Imagine a student diligently studying, preparing for an upcoming exam. Interphase itself consists of three subphases, G1, S, and G2. During G1, the cell grows and synthesizes new proteins and organelles. Imagine a city expanding its infrastructure, building new roads and buildings. In the S phase, DNA replication occurs, ensuring that each daughter cell receives a complete set of chromosomes. Imagine a library meticulously duplicating its entire collection, creating two identical sets of books. Finally, during G2, the cell continues to grow and prepare for mitosis. Imagine a conductor fine-tuning the orchestra, ensuring all instruments are ready for the performance. The M phase encompasses mitosis, the process of nuclear division, and cytokinesis, the division of the cytoplasm. Imagine a perfectly choreographed dance culminating in the separation of two individuals. Mitosis ensures that each daughter cell receives an identical set of chromosomes, while cytokinesis divides the cytoplasm and other cellular components. Throughout the cell cycle, checkpoints act as quality control mechanisms, ensuring that each phase is completed accurately before the next one begins. Imagine traffic lights controlling the flow of vehicles, preventing collisions, and ensuring a smooth journey. These checkpoints monitor DNA integrity, nutrient availability, and other factors, halting the cycle if any problems are detected. This intricate regulation of the cell cycle ensures the orderly growth and renewal of tissues, a testament to the elegant control mechanisms governing cellular life. Section 6. A Universe Within, reflecting on the wonders of cellular life. Our journey into the microscopic realm has revealed a world of astonishing complexity and elegance. From the intricate dance of mitosis to the molecular symphony of protein synthesis, the cell stands as a testament to the power of natural selection, shaping life's processes over billions of years. The understanding of cellular processes has revolutionized medicine, agriculture, and biotechnology. From developing life-saving therapies to engineering crops for increased yield, the applications of cell biology are vast and ever-expanding. As we delve deeper into the mysteries of the cell, we gain a profound appreciation for the interconnectedness of life. Every organism, from the smallest bacterium to the largest whale, is composed of cells, each carrying out the fundamental processes of life. The exploration of the cellular world is a journey without end, a constant source of wonder and inspiration. As we continue to unravel the secrets of the cell, we unlock new possibilities for understanding ourselves and the world around us.